everyone, my name is Natalie Asmus and this is my final project on prion disease. In this presentation, I'm going to cover both the genetic and neurological etiology of the various forms of prion disease, including how the disease affects the brain, the underlying cause behind various symptoms and signs, and finally the future of treatment and what you can do to learn more. First, it's important to understand what a prion is. Prions are cellular glycoproteins, typically found on the surface of the cell, most commonly among neurons or other nervous system tissues. To the right of your screen are two models of the prion protein in its folded three-dimensional shape. In its natural state, the prion protein, referred to as PRP, also has a variety of important functions, which we will cover in the next slide. However, as we will later learn, prion disease involves a mutated misfolded form of the prion protein. To distinguish between the two, scientists refer to PRP as PRPC in its natural state and PRPSC to denote the mutation. The gene responsible for the synthesis of PRP is PRNP, located on chromosome 20. However, there are other genes such as PRND and SPRN that are members of the prion protein family, along with a hypothetical fourth gene, PRNT, located nearby. These genes produce proteins that display similar mechanisms of PRNP and often contribute to the development of prion disease. The lack of obvious phenotypes associated with PRPC in mice has made it difficult to determine the specific functions of the prion protein in its normal state. However, studies have determined that it is capable of binding copper in vivo inside the womb, an important trace element involved in the formation of red blood cells, glucose and cholesterol metabolism, and the synthesis of critical proteins and enzymes, among other functions. The prion protein plays an important role in the development of the nervous system. It also protects neurons from degradation, acting as an inhibitor of abnormal apoptosis, pre-programmed cell death. A 2009 study in Nature suggested that the expression of the prion protein in the olfactory bulbs could also be linked to a role in odor detection. Evidence from Dr. Le Pichon, whose paper demonstrated that prion protein knockout mice displayed impaired odor-guiding behaviors, such as finding buried food and determining between scents, further supports this theory. Finally, researchers at Columbia University and the Stowers Institute for Medical Research in Missouri have also revealed the importance of the prion protein in long-term memory formation. Focusing on a protein known as ORB2A in fruit flies, or CPEB in humans, scientists displayed how prion's self-perpetuating chain reaction can maintain synapses for extended periods of time and help to stabilize neural connections. Now that we understand what prions normally do, we'll focus specifically on the mechanisms by which they go haywire and the resulting effects. Prion disease is a deadly neurodegenerative disorder characterized by a conformational change in the prion protein, from PRPC to PRPSC as we discussed before. The cause for such a change can be due to a variety of factors, some of which are still relatively unknown, and notably alters the protein's characteristic structure. PRPSC proteins fold abnormally and form aggregate clumps, which accumulate in the brain and lead to the breakdown of neurons and other brain cells, much like the amyloid beta clusters characterized by Alzheimer's disease. Because the disease has a notable incubation period during which the misfolded proteins begin to recruit normally folded proteins and continue to replicate, symptoms do not appear until the disease is in its late stages. Many forms of prion disease exist among both humans and animals alike, and while extremely rare, there are only around 1 million cases annually worldwide and around 300 per year in the US. Prion diseases are always fatal and display rapid deterioration, sometimes within less than a year. Here we have a side-by-side comparison of normal PRPC next to the mutated form of PRPSC. As previously mentioned, the conformational change alters the structure of the prion protein, as amino acids switch from an alpha to a beta helix configuration. Because structure is so critically linked to function, this mutation alters the corresponding role of the prion protein. Even though prion disease is extremely rare, there are certain factors that can increase your risk. Having a family history of the disease is almost a certain indication, given the genetic form of prion disease is caused by an autosomal dominant mutation, meaning it is often in the family's best interest to receive genetic testing. Additionally, prion disease can spread through direct contact of infected tissue, so eating meat from animals infected with prion disease, often referred to as mad cow disease, receiving a blood transfusion or medical graft from an affected individual, or having an operation with contaminated medical equipment can also put you at risk, even though the likelihood of these events has drastically decreased with recent safety implementations. There are three large categories of prion diseases among the various forms that exist. The first and largest category is made up of sporadic prion diseases. Around 80% of all cases are sporadic, most of which are diagnosed as Creutzfeldt-Jakob's disease, the most common form of human prion disease. While the exact cause of sporadic cases remains somewhat of a mystery, sometimes scientists have proposed that somatic mutations, which differ from typical inherited genetic mutations and that they are acquired by a somatic or body cell and are only passed on during subsequent cell division rather than from one generation to another, may be one contributor. Symptoms of sporadic prion diseases such as Creutzfeldt-Jakob's disease typically appear between the ages of 45 and 75 and are characterized by rapidly progressive dementia. As previously mentioned, these cases are fatal, and most run their course within a single year. To illustrate the rapid progression of brain deterioration in someone with Creutzfeldt-Jakob's disease, here is a series of MRI scans taken only 7 months apart. Frames A and B display the initial brain structure, and by frame F, you can see just how much of the brain tissue has deteriorated in little more than half a year, showing how aggressive and deadly misfolded prions can be. Next, we will focus on genetic prion diseases. Accounting for around 15% of all prion disease, these forms are significantly less common than sporadic cases and include variants such as inherited Creutzfeldt-Jakob's disease, Gershman-Straussler-Scheinker disease, or GSS, and fatal familial insomnia, FFI. Collectively, the average age of onset symptoms occurs between age 30 through 60, and patients often have an average lifespan of 2 to 5 years from diagnosis. However, each disease is unique, so it is often impossible to predict with certainty how quickly the disease will progress, even with genetic and blood tests. Genetic prion diseases are caused by mutations to the aforementioned PRNP gene, shown here at right. Most scientists agree that the change in PRNP is a gain-of-function mutation, meaning the protein evolves to serve a new role. Unfortunately, genetic prion disease is caused by an autosomal dominant mutation, meaning that it takes only one copy of the mutated allele to display symptoms, and that there are no carriers of the disease. Surprisingly, while a family history is often a grim indication, the mutation has to start somewhere, meaning that around 60% of those diagnosed with some form of genetic prion disease are the first in their family. Finally, although some genetic conditions display various degrees of penetrance, the degree at which a given genotype displays a characteristic phenotype, or the proportion of individuals with the mutation who display expected symptoms, all documented cases of prion disease thus far have been completely penetrant. Along chromosome 20, shown in this diagram, there are over 40 different sites where a mutation can occur. These include both nonsense and missense mutations that involve the addition or deletion of specific amino acids within the polypeptide sequence, along with substitution mutations that simply swap out one amino acid for another, without changing the codons of other triplets in the sequence. The last broad category of prion diseases includes CRU and iatrogenic Creutzfeldt-Jakob disease among humans and bovine spongiform encephalopathy, BSE or mad cow disease, and scrapie among animals. Although these acquired forms of prion disease have been the cause for widespread panic in the past, thanks to new safety regulations, today they are extremely rare and account for less than 1% of all cases. Although you cannot catch prion disease like you catch a cold or the flu, these acquired forms can be spread from human to human, or even animal to human, via direct contact with infected brain tissue. This includes blood transfusions and consumption of meat from infected animals, among other circumstances, as misfolded proteins invade lymphatic tissues and travel to nerves from the gut in the autonomic nervous system to the spinal cord and the brain. In the late 1950s through the early 1980s, before human growth hormone could be made synthetically, many children in the United Kingdom received injections from cattle that were unknowingly infected with mad cow disease, or BSE. In recent decades, a majority of variant Creutzfeldt-Jakob disease cases in the area have been traced back to those injections. Another notable case of acquired prion disease involves the Foray people of Papua New Guinea, shown here at right, who spread the disease among their tribe via cannibalism. While this problem has since been eliminated, there have been a lot of really interesting books and documentaries on the subject, which I suggest you check out if you're interested in finding out more. Now that we have covered the various types of prion diseases, it is important to understand exactly how these misfolded proteins affect the brain and cause such serious damage. According to scientists at the National Institutes of Health, in many cases of prion disease, prion protein often relocates to the cytoplasm rather than residing on the cell surface as it should. Inside the cell, prion protein binds to another protein known as mahogunin, believed to be essential for the survival of some brain cells. When prion protein binds to mahogunin, it deprives neurons of existing mahogunin and induces cell death. Studies published in the journal Cell indicate that in mice infected with prion proteins, PRP formed aggregate clumps with mahogunin, and because of the subsequent cell death and neural degradation that followed, patients often developed tiny holes in their brain tissue, leading to the spongy-like appearance that is characterized by bovine spongiform encephalopathy and other related disorders. Researchers have determined that PRP likely interacts with other proteins as well, but that mahogunin is especially important. Mahogunin plays an important role in ion and protein binding, cleaving proteins involved in alternative splicing, and the negative regulation of G protein and cyclic AMP signaling pathways. It also plays an important role in targeting misfolded proteins and promoting degradation, preventing the accumulation that destroys brain cells and results in neurodegeneration in the first place. What you see here is the characteristic spongiform encephalopathy of prion disease. The misfolded proteins form clumps that inhibit cell signaling and synaptic transmission, meaning brain cells die out and degenerate, leaving behind holes and creating an appearance of spongy brain tissue. This image compares a sample of normal brain tissue stained with dye to brains of those affected by Kuru, Creutzfeldt-Jakob's disease, and Scrappy, in order to show the degree of rapid neurodegeneration and resulting physiological effects. There is no one characteristic phenotype of prion disease, but rather a culmination of many debilitating signs and symptoms that arise as one's condition worsens. Accumulation of misfolded proteins, as we've just seen, results in cell death, which then leads to neurodegeneration or loss of connections between neurons, meaning messages cannot be transmitted to and from the brain and spinal cord. Malformation of various brain structures driven by insidious proteins often results in seizures, memory loss, and progressive dementia. Damage to the front and middle sections of the frontal lobe can also drive personality changes, wherein the patient suddenly becomes apathetic, disconnected, or displays socially inappropriate behavior. The frontal lobe controls decision-making and planning, so when inhibition is impaired, a patient may display elation or aggression for no clear reason, or seem to be overly confused or inattentive. In addition, damage to the cerebellum, basal ganglia, brainstem, nerve fibers, and neuromuscular junctions, all of which play a role in coordinating muscle movements and relaying information from the brain to the muscles, can result in dysarthria, an inability to normally articulate words. Although dysarthria may seem like a language or communication problem, it is actually a motor issue. Neurodegeneration can damage nerve fibers, which connect the cerebral cortex to the brainstem and relay information that helps control and coordinate the muscles used to produce speech, including those of the lips, tongue, and vocal cords. Other motor problems, such as ataxia, can also arise due to the damage from prion proteins. Ataxia is the result of degeneration to and deformation of the cerebellum, whose role is to coordinate voluntary muscle movements and to help maintain posture, balance, and equilibrium. As both prion disease and symptoms of ataxia worsen, muscles become less and less responsive to corresponding commands from the brain, affecting balance and coordination, often accompanied by muscular tremors and jerks. Those with prion disease may also have trouble recognizing or perceiving the world around them. Some develop cortical blindness, a condition mediated by damage to the occipital lobes, responsible for controlling our visual system. no damage to the eyes actually occurs, but instead, the processing of visual information is disrupted, and as a result, the brain is not able to accurately interpret what the eyes see. One important diagnostic distinction of this condition is that the pupils will still respond to light, indicating that there is a problem with the neural processing of retinal information rather than a problem with the eyes themselves. Although prion diseases are incredibly rare in and of themselves, there is growing evidence that they may in fact share similar features with more well-known neurodegenerative diseases as well, including Alzheimer's, Parkinson's, and Huntington's. And although some controversy remains, a landmark study conducted by Dr. John College at the University College London found surprising clumps of beta amyloid proteins while studying the post-mortem brains of several patients between the ages of 36 and 51 who had died of Creutzfeldt-Jakob's disease after receiving initial tainted human growth hormone injections as children, which was previously discussed in acquired prion diseases. These patients displayed signs of Alzheimer's significantly earlier than would normally be expected, suggesting that the injections may have carried small traces of beta amyloid proteins, displayed at the bottom right-hand corner of a brain imaging scan that shows the neurodegenerative effects of Alzheimer's, and that Alzheimer's may share a similar etiology to prion disease. This is especially exciting because if proven to be true, it is possible that many other neurodegenerative diseases could share the same basic pathology and mechanisms, meaning future treatments could target one and all. Additional evidence published by Stanley Prusiner, the Nobel laureate who discovered prions in the 1980s, also suggests that one prion protein may be responsible for several different diseases, and although other studies have since been unable to replicate the same results, it is exciting to think about the potential implications and progress that could soon be made. Currently, however, there is no viable treatment or cure available for prion disease, and all cases are eventually fatal. This is not to say that research is not ongoing, and there are a handful of medical and scientific institutes around the world that are actively engaged in development and identification of a potential cure. Progress has been made in recent years, and scientists have also begun to rule out many options through trial and error. In the next few slides, we will discuss these various novel treatment approaches. However, it is important to keep in mind that many are only in the very beginning stages, and it is unlikely that any real prospects will be available for at least another 15 years. The first treatment approach we will discuss is antibody therapy. These proteins produced by the body's immune system typically bind to foreign invaders, including pathogens, bacteria, and other proteins. However, antibodies can also be engineered outside of the body and injected into patients to treat diseases such as cancer, as we will see on the next slide. Past experiments using antibodies against prion proteins have proved successful, but there are still barriers to human applicability. Back in 2001, antibodies injected into infected cells in culture were shown to wholly eliminate the misfolded prion protein, while another study, which you can find out more about by clicking on these hyperlinks, done with mice a few years later yielded similar results. However, these prions were injected peripherally, meaning outside the brain. The mice were cured of the disease before the misfolded proteins could even reach the brain, but died just as often when PrPSC was injected directly into the brain, or the disease was not treated until it reached the brain. Furthermore, antibody injections present an additional challenge due to their large size and inability to cross the blood-brain barrier. Nevertheless, ongoing efforts to develop ways to successfully inject antibodies through the brain rather than through the bloodstream carries potential for the future. Here is a simple model of how antibodies are currently used to treat diseases such as cancer. When the cell cycle goes haywire, promoter oncogenes are permanently activated and send signals to activate receptor proteins along the cell surface. Antibodies can block this signal in one of two ways: binding to either the receptor surface or to the ligand signaling molecule itself, thus effectively blocking the resulting signal and gene production. Monoclonal antibodies can also be targeted to match a particular antigen presented by a specific protein, such as a receptor of a tumor cell in cancer or the prion protein in prion disease, binding to and preventing further growth. Another viable option involves gene silencing. A 2003 study published in the journal Cell, which you can find out more about by clicking here, found that when the prion-producing gene, PRNP, was turned off in cells of mice infected with prion protein, the mice fully recovered, even after the initial infection. Scientists believe that if they can turn off PRNP in humans, they will be able to stop, or at least limit, the production of prion proteins and thus treat the disease. And while the genetic manipulation used in the study I just mentioned is not available for human use, scientists are rapidly expanding their toolboxes when it comes to gene manipulation, including both RNA-mediated interference and antisense oligonucleotide therapy. The small strands of DNA and RNA used in various gene editing techniques present similar issues concerning size and their ability to cross the blood-brain barrier. But if these challenges can be overcome, the ability to disrupt the central dogma of DNA transcription and translation could open several novel pathways and potentials for treatment. Here we have a model of RNA-mediated interference, or RNAi. RNA interference begins with a double-stranded RNA that is then cut into small fragments known as small interfering RNA strands, or siRNAs. A polyubiquitin protein complex binds to each siRNA fragment, which then targets a single strand of complementary mRNA, the messenger RNA that transcribes the DNA blueprint into a reusable copy during protein synthesis. The complex targets each strand of mRNA, cleaving and integrating the genetic information to disrupt the normal dogma of protein synthesis. Without mRNA, DNA instructions cannot be translated into a polypeptide chain that will eventually be folded into a 3D protein, thus silencing the gene and protein production. Another method of gene silencing involves antisense oligonucleotides, which operate in a similar manner. Antisense oligonucleotide therapy utilizes short single-strand pieces of chemically modified nucleotides, the base unit of DNA and RNA, known as oligonucleotides. These molecules are engineered to be complementary to a particular mRNA sequence and are inserted into cells where they then proceed to bind to these target mRNAs before destroying the strand in one of two ways, either by physically blocking translation and the docking of tRNA to each codon themselves, or by recruiting an enzyme known as RNase H to degrade the mRNA. And although antisense oligonucleotide therapy and RNA interference may seem nearly identical, there are some key differences. Each recruits a different RNase enzyme to help degrade the complementary mRNA, and because RNAi molecules, the siRNAs, are twice as large as oligonucleotides, being double-stranded instead of single-stranded, it may be more difficult to apply RNAi across the blood-brain barrier, an issue we have previously discussed. The final and arguably most promising therapy we will discuss involves small molecules. Small molecules are chemical compounds like aspirin and ibuprofen, most common drugs you can probably think of. Their size and corresponding ability to cross the blood-brain barrier makes them a more likely candidate for viable treatments, and there are currently several research groups actively involved in chemical tests and experiments. Scientists have already discovered 3 small molecules, as indicated by a group of studies published in 2007 and 2013, which you can find out more about by clicking here, and that have been shown to work against mouse prion strains: IND24, CPDB, and ANL138B. And while IND24 and CPDB have not been able to produce the same results in human prion strains, ANL138B is still being tested as a viable possibility, and other compounds may soon be identified by ongoing projects. Although viable treatments are still a good 15 years from the market, scientists and researchers are closer than ever before to finding a cure for prion disease, giving hope to the small but desperate population of individuals and their families suffering from these awful neurodegenerative conditions. Teams of scientists at the Institute for Neurodegenerative Diseases at UC San Francisco, the MRC Prion Unit at University College London, the Prion Center at LMU in Munich, Germany, and the Prion Group at the Scripps Research Institute in Florida are all actively engaged in a number of fascinating projects and experiments that hope to model and advance some of the potential therapies we have just discussed. You can learn more details about each institution with a quick Google search by clicking on the links I provided in the presentation over on the right. But I'd like to highlight one initiative of particular interest, It involves a collaboration between GlaxoSmithKline, a London-based pharmaceutical company, and the MRC Prion unit at University College London. In an effort funded by the Department of Health, GSK and UCL are working to identify effective small molecule therapy for the treatment of prion disease in humans. They have an assay, or experimental tests, to look for molecules that will bind to and stabilize the prion protein in its cellular form, thus preventing the protein from converting to its misfolded pathogenic form. So far, scientists have already screened several compounds in various cellular models of prion disease, and are now currently working on chemically modifying these compounds before testing them in disease models. Now that you hopefully understand more about the etiology, neurology, and future of prion disease, you may want to find out more or learn about what you can do. To increase awareness of prion disease and encourage current research efforts, it is important to get informed and get tested if you think you may be at risk. There are a lot of great resources that have been published within the past few decades, both books and scientific papers, some of which I've highlighted throughout the presentation. But I've picked out a few select titles here that I think are especially helpful. If any of these titles catch your eye, there are links within the presentation to Amazon, which can provide a brief synopsis. And although it is not specifically centered around prions, I would also strongly recommend The Dueling Neurosurgeons by Sam Keen, an excellent and entertaining book that covers a wide variety of topics within neuroscience, and is what introduced me to prion disease in the first place. Congratulations, you've reached the end of the presentation. Whether you stuck around for the whole video in its entirety, and I commend you, or just skipped around to find what was most interesting to you, I hope you learned something new and come away with a better understanding of prion disease and its underlying neurological mechanisms. I certainly learned a lot more while researching and completing this project, and hope that at the very least, you too can understand and maybe even share my fascination and frustration with this incredible and terrible condition. Thanks for watching. If you have any comments or questions, I'd be happy to try and address them.